like I said, all of the work that's presented isn't just me or mine. It's really the result of a lot of different people, and we'll be doing some, you'll see some of that in the acknowledgments. So the first talk this morning is really a method overview. So this is really the molecular section. We're going to be covering, the molecular, the bioinformatics, experimental design, pluses and minuses, all of the different things related to GBS. And I want to start off right at the beginning to tell you I'm not here to sell you on GBS. You may surprise you. I know you're all coming to a GBS workshop, but it's not always the right tool for the particular job that you have, and we'll talk about that as we go. But the whole idea here with this workshop is that you get a really good grounded understanding of what the molecular method is, how you can tweak it for different things, how the bioinformatics work, how you can tweak that, and when it's applicable to particular situations and not. So that's what I'm hoping you get out of this workshop. So in this talk, I'm going to talk about the background and goals of GBS, the lab protocol, a little bit of review of Illumina sequencing for those who don't know much about that, and it's really key to making the whole thing work. How I design the GBS adapters so that they work with the Illumina system. A little bit about how it's different from the RAD protocol. How you might modify it for different species, because I'm sure we're not all working on corn, which is what I started this on. And then a little bit about the kind of GBS workflow in a high throughput kind of service lab. So to start off with, when we do GBS in a large genome species, the whole idea is that we want to reduce the complexity of the genome. So we're not sequencing the entire genome, right? That would be really expensive if we wanted to do a lot of samples. What we're doing is we're reducing the complexity and focusing on a sampling of the genome. Okay, so there are a couple different ways of doing this. One would be a target enrichment approach, so you could use long-range PCR, molecular inversion probes, or like some of these hybridization-based methods like sequence capture. Okay, these things are technically pretty difficult and expensive. Another way is using restriction enzymes, and this has been, people have been using restriction enzymes to do this job for a long, long time, and it's technically much less challenging. And when we use methylation-sensitive restriction enzymes, it helps us filter out the repetitive fraction of the genome. Okay. This slide I keep in this talk because a lot of times folks think that all the action is happening in the genes, and it's not necessarily. And so what I'm showing here is a chromosomal segment where there's a QTL here. We have a start and stop codon and exons in a gene. So if you do an exon capture-based method, you'll get information about what's going on in those exons. But if, in fact, the action is happening upstream in some regulatory region, like we see in these particular examples in maize, you'll never actually be right where the action is. But if you do something like GBS and you map a large number of markers genome-wide, you have a lot better chance at really getting something that's tightly linked to the causative polymorphism. Okay, so what did we do? We created this public genotyping informatics platform based on next-gen sequencing. And so the first thing I did was work on creating this protocol, right? It took me a good long time to create something that I can show now in one slide, and you'll see that in a bit. And we do either low or high coverage sequencing. This slide's a bit old because we were using the Illumina GA or the GA2 in the early days. Now, of course, we're using the HiSeq 2500. And then we developed informatics pipelines, which we use to anchor markers across the genome. We impute missing data if needed, and then we combine the genotypic data that we generate with phenotypic data and other things to do all sorts of things like QTL mapping, genomic selection, and genome-wide association studies. Okay, so this next slide is really, this is actually kind of why I'm here, right? So I've got a particular kind of philosophy that I think, if we do something really cool and really widely useful, we should share it with other folks, especially in science, right? So we made the method available for anybody to use and modify. The analysis pipeline details and all the code are public for people to access, use, modify themselves if they're programmers, or I can hire somebody that's a programmer. If you're working in a particular species and you use a particular enzyme and your colleague is also working in that species and you use a particular enzyme, then you actually have data set compatibility. And so talking with each other in your community is really, really important to set this up and to keep it going. of course, we published in PLOS One. That's not the journal Science, but everybody in the planet with a web browser can read our paper. And in the Buckler lab, for public projects, the genotype calls are made available, so people can download those genotype calls and use them as a community resource. Okay, so what is the basic idea of genotyping by sequencing? So what we're looking at here is 2 different samples of chromosomal segments represented. The vertical bars are restriction cut sites, and of course this is a restriction digest based method. And when you see these arrows, we call it a sequence tag. So what we're doing essentially is just sequencing the ends of restriction fragments. And then what we're doing is comparing one sample to another based on those fragments that we sequence. And so when we do this, we can call both SNPs and presence-absence markers if we get deep enough coverage. And we also identify small indels in the pipeline, but they're not scored. Everybody with me? So one thing I haven't said, and I always like to say before I get started, I really like an interactive audience. I don't mind being interrupted. So anytime you have a question, just raise your hand and give me, let me know. So I would like to just keep everybody together. Okay, so what is, and this is another kind of explanation of GBS from a different angle. It's a highly multiplex system for constructing libraries for next-gen sequencing, but what we use it, some of the major features of this are it has reduced sample handling, few PCR and purification steps, no DNA size fractionation, this efficient barcoding system which allows us at the end to assign a particular sequence read to a particular sample, which is of course pretty key. Another thing it does is it allows you to do simultaneous marker discovery and genotyping. Most of the time in other methods, you have to do this discovery phase, right? Developing markers, and then you apply them and use them as genotype to do the genotyping. There's an inherent problem in that system, which is that you are limited in the markers that you have by the panel on which you used to do the discovery, right? And so if you go far away genetically from that panel, your markers may not actually be all that useful. They may not work, and I'm sure some of you have seen this. With GBS, the more diversity you put in, the more markers you get out, which is actually kind of a nice feature. And it scales really well, and you'll see we've got some examples of a project with 2 samples, and the Maze project in Ed Buckler's lab, which is now up to 50,000 samples. So this thing can work for lots of different sizes of projects. Okay, so to really understand the system well, you need to kind of get a good feel for how we designed it and why we designed it in the way we did. For this system, we have 2 sets of adapters. And so we digest the DNA, and then we ligate these adapters on the ends. And incorporated into these adapters, what I call the barcoded adapter, it has this P1 Illumina sequencing primer 1 site. So you can imagine that I looked at what Illumina did and how their system worked, and I used bits of that to build this system. That's exactly what I did. And then it has a barcode, and it's 4 to 8 base pairs long, and that's significant, and I'll talk more about that in a bit. And then it has this common adapter with Illumina sequencing primer 2. And when I designed this system, I wanted to have maximum flexibility, so this system can do paired end reads, but we don't often do that. It turned out that it's not all that useful, but it's built into the system if you want. And there's a couple different enzymes that are commonly used at the Institute for Genomic Diversity at Cornell, and the thing that's in common with all of these is they leave sticky ends, and so you design these sticky ends to be complementary to the overhangs that the restriction enzyme leaves for nice ligation. Okay, does that make sense to everybody? Okay, so here's my one slide on actually what you do in the lab. It took me about 1.5 years to work this all out. What we do is we plate the DNA and dry it down, and you plate the adapter pairs, and in each one of these 96 wells has a different barcoded adapter so that you can sort things out later. And then you dry that down. And the reason that you do the dry down step is because for the next steps you want to be able to add the same amount of liquid in each well to make it really easy. And so what you do is you make a master mix of the restriction enzyme, whichever one you're using, and you aliquot it in every well. and you incubate that for the appropriate amount of time, so it does the digest. And then you make another master mix for ligation with the ligase, and you put it right on top, so you're not moving anything. And then you can conduct a ligation. You pool aliquots from each well into a cleanup step, just a QIAGEN column. You clean it up, add some primers, do a PCR, do another cleanup, and evaluate the fragment sizes on an Experion or a bioanalyzer. And I'll have many, many more examples of those later so you can see what they are. The key thing that, for me, when I was working on designing the system was to make it have as few steps as possible, right? The few possible steps I could, and also to not handle things very much. Because every time you start moving things around, you have an opportunity to introduce an error, right? So this system was designed for massive, massive high throughput, and you don't want to have to fiddle with things if you can help it. Okay. Now, one of the things that can happen is that the adapters can just ligate to each other, and that produces a dimer, right? And that dimer has all the component parts needed to actually attach to the flow cell and be sequenced. But we know what that information is. It's not helping us scientifically at all, right? It's not adding anything new. So for each species enzyme combination, you do this titration step, and it's in the supplement to our +1 paper. And basically the idea is you hold the concentration of DNA constant across 8 samples, and that's convenience for a strip tube, right? Those of us that work in the lab. And then you change the amount of adapters that you put in, and then you run through the process, do the PCRs, and then run them out. And when you get the right ratio of adapters to genomic DNA, that dimer disappears. And then you know what the amount should be. And from then on out, you just go. Does that make sense to everybody? So this is really simple. So a lot of times you'll see people with protocols that will say, well, okay, yeah, we always get these dimers, and so we're going to we're going to put it in a gel, and then we're going to cut the gel, and then we're going to purify the gel. I don't like to, my first project with DNA sequencing technologies was in the GA days, and I had to do a lot of that, and it's really, really tedious. And when you do it for a couple of years, you never ever want to do it again. So you sometimes get a little clever and say, what can I do to not have to do that, right? And so that's how this bit came about. Now, one of the things that happens in the preparation of GBS libraries is that we say there's no size selection step. There's none that like a gel or a pippin prep or anything like that. But the PCR step actually enriches for smaller fragments. So if you take a look at the reference genome and do a virtual digest and say, what are our fragment sizes? What would they be? This is the distribution in blue. Okay, but when we actually do perform GBS in the lab and we look and see what our fragment sizes are, it's enriched for these smaller fragments. Okay? And if you notice on the right, there's this greater than, what is that? 10,000. So APK1 leaves a lot of big pieces, but we don't see that in our sequence output, and I'll explain a bit more about that in a minute. So I've been talking a little bit about the molecular method, but what is GBS, right? It's a genotyping system. So if you have a sequence-based genotyping system, you want to make sure that the number of reads you get per sample, the number of sequence reads you get per sample, is relatively even, because what you're trying to do, in essence, is compare those one sample's reads to another, right? And if they're wildly different, you're going to have some samples that have just a few reads, and you're going to get very few markers out of that, right? I mean, that's pretty obvious. So one of the things that we worked really hard to do is to get this read even. And one measure of evenness is the coefficient of variation, which is just this standard deviation divided by the mean. And when I guess it was 2011 or 2012, when the Davy et al. Paper was coming out, which is a review, and I'll have a slide from that paper a little bit later, looking at these kinds of methods, these sequence-based genotyping methods. I took a look at that and took a look at some of the papers that he was citing, and I was looking for this number, right? What's the number in these papers? And what I found was either they didn't report the number, right, which doesn't bode well, or it was something like 0.8. That's a huge difference. So I should say that the smaller the number, the better. That's to get everybody oriented. Okay, sorry. So if you've got and this is in a 384-plex with a mean count per sample of about 500,000, and a CV of 0.22 is, we consider, really, really a nice number. Yes?
What would be the range that you would consider acceptable?
Well, that's a good question. I think it depends on the person. When I was working with Ed, I spent 6 months him saying, get that number smaller. And I didn't know what small enough was until he finally said, okay, this is good. You know? I don't think, honestly, I'm not sure he knew what exactly he was, but I think he was just pushing to see how low you could get it. I'll tell you what goes into making this number small in a bit, because I had to spend a lot of time figuring out how to do it, and so I have a pretty good idea how it works. Okay, so this is a little bit about Illumina sequencing. This is a flow cell. They don't look like this anymore, but they sure did when I started. On these flow cells, there's 8 lanes, and this is just kind of a cartoon blow-up of what's on those lanes. So there's a lawn of oligos. They come in 2 flavors, and you'll see how that works. And the whole system is based on kind of a solid phase PCR, which is an idea that goes way, way back. What you do is you take your library, and this is really an important point to get, and it can sometimes be a bit confusing. So we have this tube with PCR product, the library, right? The first thing you do is you denature it. So you end up with, in your tube, a bunch of single-stranded oligos. That way, these oligos can bind to the oligos on the flow cell. So these are complementary, and that's added on during the PCR step, okay? And then you do this thing called bridge amplification. So it just reaches over and makes another copy and makes another copy, and you get a lot of copies. And then you do a cleavage step, and you end up with what they call a cluster of essentially a clonal cluster. So each of these clusters started out from the same molecule, right? And so they're all in those clusters, all those molecules are the same. And so you may wonder, why in the world would you want to go through all of this hassle, right? Well, it really comes down to the way that the sequencing chemistry works. And Illumina chemistry is a sequencing by synthesis. So you're adding a base on that has some kind of fluorescent tag each step. And then you're shining a laser on it, and you're essentially taking a picture to see what color that is, right? Well, it turns out that just a single strand doesn't produce enough light to be detected very well. So what this process is, essentially, is a signal amplification process, so that you can really get a good signal and capture that image and make a base call. Okay, so this is basically how it works. We have a sequencing primer 1. It binds to the sequencing primer 1 cut site, and as I said, you do sequencing by synthesis. So there's your primer 1. So you add this, the complementary base, as we all know about how this works. And there's an extra bit on this nucleotide that prevents just a runaway effect where it would just fill it in. So you put that there, you excite it with a laser, you take a picture, and you say, okay, that one's a T. Okay? And then you have to wash that stop bit away so that it's available to add another base, and then you add another base. And then you just carry on through the process. And you do this, depending on the model and how you're running it, hundreds of bases. Okay, and this is an image from a GA2, a colorized image of what a flow cell would look like at just one specific snapshot in the sequencing. Okay, so that's kind of what you'd see. Now, these days, Illumina has really done a nice job of crunching a lot more of these together, so this would be substantially denser. And that's why we can get so many reads out of the system these days. Okay, so here's another concept that needs to be explained so you understand why I built the system the way I did. So what we're looking at here is one of those clusters, and in theory, you would expect everything to be the same at the same time. You add another round of chemicals, and it just goes next one, next one, next one, and then as long as that works, then you can get a good base call, right? But sometimes, as in life, things don't always work the way you'd hope. And you get this kind of situation where some are ahead or behind. And when you have that situation, you get an N, because it doesn't know what it is. It can't sort that out. So this is called phasing. Now, Illumina system actually can correct for some of this, so they do some calculations to correct for phasing issues. And I'll explain why that's important after I show you how the barcoding system works. So here's the Illumina barcoding system, and here's the GBS barcoding system. So in the Illumina system, you do 2 reads. Okay, you read your insert DNA, and then you get a file with that, all of your sequence and your big FASTQ file. And then you do another read, and you get another file that has the barcode information. And you say, well, how do you put those two together? Well, they have this XY coordinate system in these little lanes, and they keep track of where they identify where the clusters are, and they say, okay, then for each read in the sequence file, it has that position information in both cases. Now, that system doesn't always work perfectly. So there have been reports of having about 30% of data lost by not being able to match these two things up. Okay, and it has another problem for me, which is I don't know that I'm really confident that this came from the same place, right? But with the GBS system, what we do is we have sequencing primer 1. The first thing that happens is it reads the barcode, which is attached at the restriction cut site to your insert DNA. There is no second read. It comes right off in the first read. And we can be pretty darn confident that that barcode goes with that sample because it's physically attached. Okay. Now, I talked about phasing and said that there's an issue around phasing with the system. So the first 12 nucleotides in your read are used to calculate phasing. Okay? And the algorithm that does that and identifies clusters and a number of other things at that stage assumes a random or even nucleotide distribution across the flow cell. So if you took all of the things at that read, at that position, and you would expect 25% of each of the bases, roughly. Okay. Now, imagine you have a barcode or a set of barcodes that are all the same length. What do you read after the barcode? The restriction cut site, right? And that's invariant, right? So you've totally blown the assumption for these algorithms. And this is something I learned in the early days as I was trying to design this, and things just were like complete junk coming out of the system as I was going along. And then I did some investigating and found out about this, and I said, oh, well, what can we do? Well, we can modulate the position of the restriction cut site such that we don't have that issue of uniformity across all of the clusters. And so that's why we have these variable length barcodes. Is that clear to everybody? Okay. So there are a number of other things that go into making a barcodes. It's a non-trivial exercise, and I've got a slide that covers all of the bits shortly. But this is what happens when what it looks like when you look at the just a graph of the signal intensity starting with base 0 and moving out when you have invariant GBS barcodes. So you get this big, massive dip in signal intensity, which means you're getting really crummy data right here. And essentially, your run is going to fail. You're not going to get much, if anything, out of that. This is what a successful GBS run looks like. It's still a little funky compared to regular sequencing runs, but it works. This is acceptable, and it works really well. So, but I just wanted to illustrate the differences. You can see this, right, when you make a graph. Okay. So I promised that I would tell you a little bit more about barcodes, and it took a while to sort out all of the different considerations that we needed to put into this to make it work both in the chemistry and in the sequencing machine and downstream bioinformatically, because we wanted to take that into account. And I was doing this work before we even had a bioinformatics pipeline, but I was thinking, what can we do to help make it easier on the programmers down a little bit later, and also how to make it less error prone. So the barcodes are specific, enzyme specific. That should be relatively obvious to the molecular biologist because the overhangs are going to be different. So you've got to have enzyme specificity, and they must not recreate the enzyme recognition site. So you remember when we went through that one slide that just showed you the molecular biology, and it was just really two easy master mixes, right? Well, in those tubes are the barcoded oligos, right? And if the barcode recreates the enzyme recognition site for the enzyme you're using, it's going to get cut, right? So we don't want to do that. So we avoid that by just not doing it. The sets must be a variable length, for reasons I just explained, and the bases must be well balanced at each position, and we kind of have gone over that. And these two things happen at the same time. So when you're designing a set of barcodes, you have to consider the cut site and the length and all that as well. And they must be different enough from each other to avoid confusion if there's a sequencing error. Now, that's not the only place there could be an error. There could be an error in synthesis as well, right? Those are the two possibilities. And the way I design this system, if there's one error, you can correct it. You can say, oh, you can reassign it to the proper barcode. If there are two errors, well, you can catch it, but you can't correct it. And this bit here was actually borrowed from digital signal processing, and that's something like when you use your cell phone and you get you get a better signal than you could by having this kind of thing built into that technology, and it's the similar idea. This one is that you don't want one barcode, so the variable length, you could have one that's 4 base pairs long and one that's 5 that could nest within the other one. Do you see what I mean? Does that make sense to people? Okay. This isn't strictly necessary, except it helps with the bioinformatics, because if somebody doesn't do a great job of coding, they may actually misassign something if there's a barcode one inside another. So we just avoid that problem from the get-go. And then we have no mononucleotide runs of 3 or more bases. Now, I've been doing these workshops for a long time, and we had a lot of times people would say, hey, Rob, this problem seems like you've defined it. You've really defined it, exactly what you need. It seems like it's amenable to a programmatic solution, like something that could be coded. And I say, I don't have time, to do that. I'd love to have it done, and I had many people suggest that it could be done. Well, in the end, this PhD student in the Netherlands, Thomas van Gurp, who is a coder and a biologist, came up with a really nice website, and this is it. That is a tool for generating GBS oligo barcodes adapters based on these criteria with a drop-down list of enzymes, and you plug in how many you want, and it's a lot nicer than the early days when I was trying to do this in a very tedious fashion.
So Rob, what's just as an indication, what for the maze project of you've done like more than 30,000 or 50,000 lines, how many barcodes? I mean, how many barcodes do you have in maximum? Was it like 384 or?
308. So the question was what? The question was what was the maximum number of barcodes we had in that large project? The answer is 384. You'll see that as we go through the bioinformatics that there are a number of factors that go into assigning a particular sequence read to a sample. You don't need a different barcode for every single sample you do, because there are other criteria you can add in, like which flow cell it was in and which lane, and some other things. So the barcodes are reused. Not in the sense that you take them back off and use them again, but okay. So let me be clear on that. Okay.
So basically, GBS would work for a project where you say, I only need a few markers and a very rare cutter, and make basically thousands of plants or individuals in one lane. So you wouldn't have GBS for very few markers over lots of plants.
No, so the question is, would you have GBS for few markers over lots and lots of plants? And the answer is, this method, no. I know of a paper that should be coming out sometime soon that has a nice little trick that'll let you do that too. So but this method, no. What you're really trying to do is lots of markers and lots of samples or in a large scale. Yes?
So there are other pipelines out there that will design stupid amounts of barcodes, but this one is specifically designed for the GBS approach as well, right?
That's right. This website that I had up on the last slide. Well, now I'm going to mess myself up. Yeah, it's specific. It's specific for GBS, for this flavor of GBS. And there really are, when you think about it, especially now, there are lots of flavors of GBS, right? And I kind of started out telling you that I'm not really, and this is not a sales pitch for this method. It's an explanation of how it works to give you guys the tools you need to make good choices, right? And you're going to hear that throughout this workshop. That's what we're trying to do.
But you could write it on the barcodes if you wanted to.
Pardon?
You could write it on the barcodes if you wanted to.
Could.
You could have longer barcodes.
Oh, you could have longer barcodes, yes, but the longer, so you could go, like, if your, maybe I'm thinking your question is something like, why not have a barcode length of 12?
Yeah, that's what I'm saying.
Yeah, so, well, because we're kind of cheap and oligos are expensive. You know, this part of the method, the design considerations with this is something that's really inexpensive. So wherever I could make it less expensive, I did.
And also, the sequencing technology, you couldn't get so long reads, so you're throwing away. I don't know if we showed how you sequence through the barcodes yet, but
Yeah.
you're throwing away a lot of sequencing.
So I think now's a really great time for my colleagues from Cornell to come up since they're here, and I will introduce them.
Sorry, you need to.
As they've had the explanation of what happened to us, so no worries, right? Everybody's. So this is Katie Heima. She is at Cornell at the Institute for Genomic Diversity and what's the other? Bioinformatics Core. Yeah, the names keep changing, but the faces stay the same. Yeah, and this is Jeff Globitz, and he's in the Buckler lab. So Jeff does, well, one of his big jobs is the one that's not fun, which is managing a big project, a big grant. He's a project manager, but I think he really likes to code, and he does a good bit of that, and he'll be talking to you about that later today. And Katie actually was in the same position that you guys are at one stage. She attended a GBS workshop, and eventually became probably the person who's done more analysis on GBS of different species than anybody on the planet. So I'm really happy to have these two here to help me make sense of this stuff. So you guys can just like chill. And there's a microphone there so that we can hear you.
So I should mumble into it?
Yeah, just mumble into it, it'll be fine. Okay.
So you were just saying that because the read length is limited, if you extend the barcode, you lose
Yeah.
sequence information.
So I think that
that too.
maximum sequencing length was at 100 base pairs.
In the early days.
In the early days when this method was being designed, but there was also a significant drop off in quality at the end of the reads, even more so than there is now. So you wanted to maximize.
Yeah, you're right. That was the other consideration. It's been so long ago since I actually did this that it's forget some bits and pieces, and this is why you have friends. Okay. Yes.
Question regarding phasing. I know that you can correct Illumina phasing by running the offline base caller. Is it something that could
So that could be done.
interest?
So the question was that the Illumina base caller can be run in the offline base caller mode, and would that help? Yes, it would. But one of the main design considerations I had as I was doing this was to develop a method that you could actually take your tube of library and send it to pretty much any Illumina sequencing provider and not have any tweaks on their end, right? Because that's another thing. As soon as you start having to change things and communicate those changes to your sequencing provider, you're just opening yourself up for problems. It's hard enough as it is, and I think a lot of you know that.
I'm asking that because running the OLB can be quite long, quite pages, so I'm not sure if it's quite beneficial to do that, or if the GBS is robust enough to go on without redoing the phasing.
It is.
The other reason, though, we like to actually sequence through the restriction site to make sure that
Yep.
it's there.
And that information is used
Yeah.
later on. So, okay, I hope I didn't skip too many slides. And I'm running long, which is normal, so we'll just go with it. So, did I seem to, I don't know, we'll just carry on. What are the most significant technical issues? Well, there's two, and we're going to talk about these a lot because these are things that we want you to have so deeply embedded in your mind that you'll wake up and say, oh, it's DNA quality. That's one of them. I've got to get that right. That's one of them. The second one is DNA quantification, and you got to get that right too. And it's these two things, really. Remember I was mentioning that about that CV number earlier in this talk? DNA quality, having high quality DNA, is a big component of getting that CV number small. Accurate quantification is another, and the third component, and we'll go over this again later, is accurate liquid handling, because if you do this right and you do this right and you do sloppy pipetting, you're not helping yourself, right? You can see how all these things go together. So those are the three main things. Okay, so this is I'll just do a quick thing on GBS versus RAD, and there's another slide in a minute. So we've seen this slide before, right? And here are our reads. The RAD technology will allow you to get some of these longer segments. GBS only really works very well for things basically less than 450 base pair. RAD will let you get at some of these ends on longer pieces, and you'll see why in just a second. So this is from the Davey et al. Review in 2011, and what we're seeing here is this is the RAD protocol. This is kind of the lab work step, and this is GBS. So you can see that there's a lot in common, right? So we both do this digest, we both ligate some adapters, we both pool, but then they do this random shearing, so that's, and then a size selection and then ligate additional adapters on, and that's what lets them get at the ends of what would be longer fragments otherwise. Does that make sense to people? Okay, so, and then we both do PCR. And that's basically, that's the major difference between those two protocols. Okay, yes?
Would it be the same with double digest RAD?
So I'm actually not that familiar with double digest RAD, but hopefully, I mean, we was talking about, you actually opened up a really nice entry for me. One of the things that I'm hoping to do here in New Zealand is build a community where we can actually put together some information about what are all these different flavors? How do they work? Which ones, it's something that, I don't think I could do just on my own, right? But if we kind of work together a bit, maybe we can create a community resource that really does address all of these questions, because there are a lot of flavors of GBS out there at this stage. And it would be great for us to have some really concrete knowledge of, where this works really well here and this works really well there, and what kinds of questions are appropriate for different types of these technologies. And there's a lot out there. So, okay. Modifying GBS. So why would you want to modify GBS? Well, there are lots of reasons. Maybe you want more markers. Maybe you want fewer markers. Maybe you want to do massive amounts of higher multiplexing. Maybe you want something that's more genome appropriate that will avoid repetitive DNA classes, or other novel applications like bisulfite sequencing or other things. So you can think of this, as just a bunch of different pieces that you can change them to do the thing that you want, right? But you have to understand the whole thing first. Does that make sense? Okay. So genome sampling strategies, they vary by species, right? All these species are different. Some of the factors affecting diversity are things like the mating system, the ploidy level, and the geographical distribution, like is it an island population? Is it all over the place? So these are considerations that you have when you're thinking about genome sampling strategies. And other factors are genome size, and the size of the genome has a bearing on the size of the fragment pool, and you'll see that in a minute. The amount of repetitive DNA, and genome composition, so the base composition of the genome can affect the frequency and the distribution of cut sites for a particular enzyme, right? And then how repetitive DNA is organized in the genome reflects library profiles, and you'll see some of that. Now, this slide here is also really, really important in understanding what we're doing, okay? So this big circle is representing a genome. Doesn't matter what species, but it's just representing a genome. And when we cut and it has two parts, right? It's got a methylated part in blue, and then an unmethylated part here. And when we cut with a restriction enzyme that's methylation sensitive, we really are only working in this area, right? So if we use a 5-base cutter, we do the cutting, and we get a GBS library that makes up that kind of fraction. And this isn't, absolute. This is just to get you the general picture, okay? So it's a sampling strategy. We're sampling the genome by making a library, and that captures maybe that much in the library, right? But when we put the library on the sequencing machine, we're not sequencing all of the fragments in that library with this method. We're doing skim sequencing, so we're subsampling. So we're getting an even smaller amount of the genome that is actually sequenced, okay? So this top example here is with a 5-base cutter, okay? If you use a 6-base cutter, it's gonna have less frequent cut sites, right? Because it's got more matching that it has to do. And the size of your library is gonna be smaller in proportion. And so when you think about it this way, with a 6-base cutter, the actual sequence fragments are a larger proportion of the library than with the 5-base cutter. Does that make sense to folks? I'm going to leave this up for a minute or so. So this is kind of a key concept when you're wanting to think about how to tune GBS for a particular species. So again, in the 6-base cutter, your library size is a smaller proportion of the genome, but your sequence fragments are a larger proportion of the library. Right? You can see. You're not looking comfortable.
Yeah, I'd like to know what difference that makes. What's the outcome of those differences?
Okay, so the question is, what's the outcome of those differences? That's great. So if you want to get more coverage of more depth of coverage in your sites when you do the sequencing, if you're already doing a large proportion of sequencing of the library, you need less sequencing to get the better depth, right? And so this one here, you need much more to sequence much more than the other to get a larger proportion of the library in your data.
If for example, both of these enzymes looked appropriate for a species and there was a decision to be made between a 5-base and a 6-base cutter, what I would ask people is, okay, well, for your project and for the data analysis that you're going to do you need more markers that are sequenced at a lower depth, or do you need fewer markers that are sequenced at a higher depth? And the answer is different based on what your project is. And so sometimes it's very clear and there's only one enzyme choice that really looks good after optimization, which if we haven't talked about already, we will. But sometimes if there's a choice to be made, that can factor in.
So, and you guys will see as we go through this workshop that we talk amongst ourselves a lot and chime in, and it's just the way we are.
I could chime in a little bit there.
Yeah, please, Jeff.
Can you go back to one?
I don't know if I can or not.
Doesn't the up arrow work?
Does it? No, I'm having some trouble with that, frankly.
Oh, well,
Seriously, let's try this one.
If you use a 6-base cutter, there's fewer recognition sites across the genome, so there's fewer fragments. And then they tend to be longer, so that makes even fewer fragments that are in range of GBS. So overall, you're sequencing fewer fragments with the same sort of overall depth of coverage, so you get more depth per fragment.
Does that make sense to people? Yeah.
These are both pretty much randomly distributed across the genome. There's no.
So here's the thing, right? Which genome? Which enzyme? Right? We're going to go into a lot of these questions as we go, because what you're going to find in this workshop is that there are the answers many times to the questions that you're going to ask begin with "it depends," right? And so this stuff is actually not trivial, right? And I'm an empiricist, so a lot of times I'm like, well, let's try a well-designed pilot study and see what comes out of it for an enzyme species combination. And that actually leads me into the next step. So, yeah.
So just there was a question on your last slide. So the proportion of your libraries that you're going to sequence is always the same fragments if you repeat it for different individuals, or?
So actually, so the question is, if you repeat if you go, let me rephrase this and see if it'll work for you. So if you go back into the same tube that you have your library and you put that on the sequencing machine, are you going to get the same fragments back? And the answer is no, because you're pulling out a random sample of that, right? You're going to definitely have overlap. You're going to have overlap, but it's not going to give you the same fragments every time. And that's actually can be pretty useful. So you could make a 384-plex library, right? And you could run it, get a lane of sequence data back, and analyze it and say, geez, I need some more sequence, right? You don't have to go make a new library. You just go back to that tube and put some more on, and you get some more sequence. So that's a concept that I wanted to bring out. Does that address your question?
Yeah, so it means that if you use a longer cut size, you're going to have more chance to sequence always the same.
You'll have a higher, yeah, that's right. You're going to be moving in that direction.
And that in turn leads to more depth per fragment.
Yeah, so the question was, if you use a longer cutter, are you going to have a higher probability of sequencing the same fragments? And the answer is yes, you'll have a higher probability.
I was just going to say, it's a rarefaction analysis. The more times you sequence, you're plateauing out to some fixed point, which is all the cut sites you possibly have in the story.
Yep, that's right. Okay. So these are some of the species that have been used, analyzed at IGD. There's a bunch of them here. I'll show you some more pretty pictures in a little while. Here's some looking at flowering plants with just a list so that things fit, because otherwise you have these thumbnails that are so small you wouldn't be able to see what it was. And then here's some things in the that are not flowering plants. Okay, so Sharon's put together these slides, some generalizations. Okay, so they've done the previous couple slides are they've done a lot of different species, right? And so what did they learn from doing all these different species? Is there any kind of generalization that that could be made based on their experience? And APEK1 works well for grasses. That doesn't surprise me in the least, because we chose that one because it worked well for grasses. That was what I developed the whole thing on, to work in corn. So not entirely surprising. PST1 seems to work pretty well for most vertebrates. And these are, again, Experion traces. These are what the libraries look like, the fragment size distribution, the fragment distribution. So some examples of that as well. And the thing is, it doesn't, the generalizations don't always hold. And this example, and we were just talking about it again the other day, that this Toulouse goose just didn't really work well with anything. And this isn't saying all birds, it's this one, right? And I was still still at Cornell when this came through, and when this thing hit my desk, I looked at it and I said to the technician, I said, I think this is ladder contamination. Somebody goofed up in the lab when they ran this thing, because look at how nice and even those spikes are, you know? I said, oh, let's do it again and see what we get. Well, we did it again and we got the same darn thing. And Sharon and I sat and pondered and talked about this, and well, I think it's some kind of a tandem repeat that's showing up with that has a cut site in it sometimes. So one of the things when you're evaluating these profiles is you're trying to avoid having these massive peaks, because what that's telling you is that you probably are actually capturing repetitive elements. And repetitive elements aren't particularly useful because they're hard to place, right? By their very nature, they're all over the genome. So if you're doing a reference-based pipeline, or even a non-reference-based, they're hard to place, and you get many, 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 many copies of the same darn thing. So it's not, your information to noise ratio is atrocious with something like this. So this is before sequencing. This is the stage where you can, when you're doing your optimizations and looking at things for your particular species, you try the different enzymes, you run the titration experiment like I described before, and then you look at these and you make some decisions. And this one is probably the worst I've ever seen. But it gives you the idea, right? That's what I'm trying to do here. Give you an extreme example that you can kind of get the idea. Yes?
Did you actually sequence it?
I don't know that it was. Do
I
you?
think they finally gave up on this one.
There you go. So the question was, was it sequenced? And Katie's response was they gave up on this one. And this wasn't the only enzyme that was tried. It was just, this was a recalcitrant species. You know, I
But this
hope
is-
yours aren't that way.
As far as I know, since I've been at Cornell, this is the only thing that has completely failed. We do get things that look better than others, but if there's something that's marginal, sometimes we'll go ahead and sequence it, and a lot of times we get perfectly good usable data from that. You might have less data than you'd like because, like Rob said, you're going to be throwing out a lot of data that's not that informative for your research question, but usually it doesn't completely fail like this.
Yeah, this is the extreme.
What do you mean sequencing?
Not
There's
that I
no
know
point.
of.
Yeah.
Yeah. Okay, so what's the most frequently asked question for new species?
Will it work?
Of course it's going to work, except for that one recalcitrant goose. It's going to work. But the question always is, how many SNPs will I get? And I warned you about this answer, and you're going to hear it a lot. So it depends. It depends on the genome size and expected heterozygosity and the amount of diversity you're sampling, and do you have a reference genome or not? There are lots of factors that go into how many SNP sites am I going to get. And you know, this is where some small-scale pilot studies can help, but well-designed experiments. Don't just toss stuff in a plate and send it off. Think about what your question is, and Katie's going to talk a lot about this as she goes through some of her talks. I will too. So this table was put up because people always want to know the answer to this question, right? And you don't really know, but this gives you some idea of what people have seen in some of the work that they've done. The species with the asterisks next to them don't have a reference genome. Okay, so some of you will have species that don't have a reference genome. So, and you see there's different ranges of enzymes in maize. So, well, the sample size is bigger now, and you know that's a moving target all the time, but just over 1 million SNPs. And 60,000 in rice at this 850 samples. Now, of course, you know that number could change if they added more diversity, right, or whatever. All those factors that I said affect the SNP count. These are just kind of ballpark indications, but not absolutes. So there's some for grape. Remember I told you that we have this big range that you could work with as far as sample number. So we've got 33,000, but the number's now 50 in maize. And then there's this verticilliflorum that had just 2 samples, right? And they got 10,000 markers in this thing. Yeah. Pretty cool.
What would you anticipate for bacterium?
Well, it depends. The question was, what would I anticipate for bacterium?
I would anticipate that would probably be a better use of your resources to just sequence
To
genomes.
to do whole genome sequencing, yeah, most of the time. So then there are strategies for doing that, because those things are small, right? So there are strategies for doing that. And so this is an example here of us telling you that GBS isn't suitable for every question, right? So you'll hear that as we go along as well. Okay, we're going to talk about this a lot throughout the workshop, but one of the, I think we used to say, Jeff, what was your saying on this one? Missing data is free?
Yeah, you get the missing data for free. Yeah.
So, and there can be a lot of it, so it's good value. So it's a part of the way we do this is what we're looking for. You'll hear about how it was designed in what's being done with this kind of thing in maize, but the idea was to do really, really low coverage skim sequencing. So that means that there are a lot of sites per given sample that just don't have any calls. And you'll also hear a lot more later about when that matters and when it doesn't matter. So we'll talk about all of that stuff so that you have an idea. But the reason it's in this session is for these points here. So there's imputation, and there'll be some discussion about that. Actually, quite a bit of discussion about that later. But what can we do in the lab? And this is the molecular end of the workshop, this part, right? So, well, you can reduce the multiplexing level so you get greater depth of coverage per sample. That's one thing. You guys does that make sense? Okay, If that's making sense, then I think we're all on the right track. You can sequence the same library multiple times, which is effectively the same as that. Or you could use a molecular option, like using a less frequently cutting enzyme. So are you guys feeling reasonably comfortable with what we've covered so far this morning on this? So, okay.
Can I ask a question? Can I go back one step and ask a question on the end?
I can't go back a step, but I'll answer your question.
Question then. So if I work with a species where there's a lot of integrations from wild species, you have slightly different repetitive elements. Do I settle for one enzyme that just has to work across the board, or do I swap and change enzymes?
Is that something that you could cover better in one of your talks later?
Yeah, let's readdress that question after I've talked a little bit, but it's going to depend on what fraction, what types of markers you want to sample to answer your question. If you want to sample markers that are from introgress regions and you don't think that you're going to capture those with a single enzyme, then certainly you might have problems answering your question. But if you realize that you're going to be missing markers that perhaps, because you choose an enzyme that works well for markers that are conserved across those species, if you can use those markers to answer your question, then it's perfectly fine. So again, it's one of these things where it definitely depends. It depends on what your question is. As long as you know that there's a potential bias there, you're a step ahead of most people.
Okay. So, and this is another thing that will happen throughout this workshop, and this is really great. Thank you for the questions, because this tells us that we're going in the right direction. You're kind of catching the ideas, and hopefully in a lot of cases the questions that you're asking we will have will be part of other presentations, and we'll let you know when that's the case. We're not trying to keep you from asking questions. We want them, but if we've got a really nice 2 or 3 slide explanation on your question when we get there, it'll be better than answering it right at the moment. Is that cool? Okay, real quickly on this. So at the Institute for Genomic Diversity, this is a schematic overview of how their workflow works. So they have a DNA sample information that's entered into a web form. That information is then stored in what they call the HTS database. And then somebody at the IGD looks this stuff over, and it's usually Sharon, and talks about is the project make sense, and we're going to take it or not, because they don't want to do something that's just not going to work, right? They want to help.
We're actually sitting down once a week and reviewing any new projects. So if you submit a project, you'll have to wait until our weekly meeting and we sit down and talk about it before you get a response. So if you don't get a response in 2 days, and you submit
don't think that they're ignoring you, but what they're trying to do is take the time to really make sure that what you're doing is going to be helpful to you. Okay, and then if that's cool, then they say, yep, this is your project's accepted. You send them your DNA samples, then the libraries are made and the DNA is sequenced, and then it gets matched up with the information that was provided with the sample input form from the database, and gets analyzed in either with a reference genome or a non-reference genome analysis pipeline. Okay, so that's just a schematic. You'll see how, and part of the reason I have this up here now isn't so much about what they're doing at IGD, but you're going to see kind of all these bits throughout this workshop. We're going to talk about all of these things, and they all have to fit together. And I think one of the main messages that we want to get across is that the molecular biology matters, right? Information storage and retrieval and keeping track of things matters. The bioinformatics matters, and all of these bits come together. And you'll recall that when I sent out the expression of interest and application form, I was asking for these 3 skill sets. They're all absolutely necessary to do this well, okay? And that's why I asked for that, so that you guys, what we really want, in the end, is for you to be able to go back to your institutions and be able to do this successfully, right? And we want you to be prepared to be able to do that. Okay, and I think, and this is just an acknowledgement slide. So as again, these were Sharon's slides, but Ed Buckler and Sharon did a lot of the method development. Jeff and Katie did a lot of bioinformatics along with Ji-Sun, and a number of people in the lab do heaps of work to make this go. So
I'd really like to say here that Ed Buckler also did a ton of the bioinformatics in there.
That's absolutely true. Yeah, Ed did heaps of bioinformatics work.